This year's Maryland Life Sciences Bioinnovation Conference is sponsored by Avantor. From breakthrough discovery to agile delivery of mission-critical products and services, Avantor is a trusted global partner to customers in the life sciences and advanced technologies and applied materials industries. Our leading global customer channel, VWR, offers an integrated, seamless purchasing experience. For more than 23 years, we have collaborated with BioBusiness Solutions to play a critical role in helping members develop scientific breakthroughs. We set science in motion to create a better world. The Maryland Department of Commerce. Welcome to Maryland, where day by day, dreamers are becoming doers, makers, and pioneers of the future. And while the state's code breakers, vaccine makers, and next-gen manufacturers are pioneering our future, they're doing it in a place that's best for their future. Make a move to Maryland to take advantage of our natural business assets, boundless resources, and amazing quality of life. The Montgomery County Office of Economic Development. Commercialize your science near the world's top scientists at NIH and FDA. Connect with us at thinkmoco.com to find your success in Montgomery County, Maryland, the immunology capital next to the nation's capital, and the Pharmaceutical Research and Manufacturers of America. Pharma represents the country's leading innovative biopharmaceutical research companies, which are devoted to discovering and developing medicines that enable patients to live longer, healthier, and more productive lives. Since 2000, pharma member companies have invested more than $1 trillion in the search for new treatments and cures, including $91.1 billion in 2020 alone. We'd like to thank these and all of our sponsors for their support of this year's Maryland Life Sciences Bioinnovation Conference.
We're giving people who have little or maybe no options another chance at being with their loved ones.
People here are really passionate about being here.
It's really rewarding to know that the work that we're doing here is going to help change people's lives.
We literally are getting bags of patient cells who have failed a previous therapy and they need help.
What we're trying to do is teach our patients' immune system to fight the cancer.
There's not a single thing that we do that doesn't have a direct impact on the patient.
What Kite has done in locating Maryland is really strategic.
Frederick County has been absolutely amazing to be able to help us to establish this very unique facility. We're really quite happy with our partnerships with the local communities here. So Hood College, we have a collaboration with them to where we initiated a lab jointly. I'm grateful to say that we're starting the apprenticeship with Frederick Community College. And these are the kind of things that I hope we start going outside and thinking a little bit differently, and really engaging people who want to learn. And with the research coming up, it's going to be amazing where we see the site and also the industry in the very near future.
This panel discussion will focus on cell and gene therapy. Please welcome your moderator, Jeffrey Hung.
Good morning, everybody. Welcome to this morning's second panel titled Cell and Gene Therapy, CMC, and the Manufacturing Hurdles and the Solutions. My name is Jeff Hung. I'm the moderator of the panel. I'm the general manager of Vigene Biosciences and a Charles River laboratory company. I'm a PhD trained geneticist. So back in 2016, when I became one of the first employees of Vigene Biosciences, I shared the same vision as then the founder, Dr. Azarian's son, about making gene therapy affordable. And in 2016, I orchestrated an acquisition of Omnia Biologics, which is a pure play viral vector and vaccine GMP company. And since then, we really witnessed and experienced tremendous growth in cell and gene therapy area. While we enjoyed profound fulfillment, and when we see that we provide the clinical trial material to the patients, we also experience our share of growing pain and struggles in the CMC and the manufacturing of cell gene therapy. I'm incredibly excited about this world-class panel that we put together in the area of gene and cell therapy manufacturing and CMC. We thought it will be fun to offer this hour as a free consultative session to this audience on site and audience online, to be able to share our experiences and to offer you an opportunity to ask questions to this panel of world-class experts. To lay out the foundation and the framework of today's discussion, I have a quick slide. Okay. So this slide is about the gene therapy manufacturing and the analytics roadmap that I drafted, outlining just some bottlenecks and the to-do list to make gene therapy manufacturing and testing more speedy, efficient, and of high quality. We don't have all the time to belabor the detailed points on this slide, but it suffices to say there's from plasmid to helper virus production, upstream production, downstream purification, to analytical release testing. It takes a lot of things to go well to be able to release the clinical material to the patients. So that's gene therapy and the viral vector production. And this is end-to-end cell and gene therapy workflow that the Charles River offers. I use that as a backdrop to illustrate how complicated a cell therapy manufacturing is. From cell supply to viral vector plasmid supply to cell therapy manufacturing to release testing, it really takes a lot of steps and a lot of steps to go well for the CMC and the manufacturing to be able to support the patient dosing and ultimately the commercial release. So with that, I'm like to going to introduce our panel and going to introduce each one, and then we will take turn. They will take turn to share this one success story and how that story is successful. And then that's first round. And second round, we'll take turns to share a lesson learned. And then we'll open the time up to the floor and to the audience online. Okay? So I will start the first introduction based on the alphabetical order of the last name. It's my great honor to introduce David Anderson. And David Anderson leads the quality team at Gilead, Kite's newest cell therapy manufacturing facility in Frederick, Maryland. He has a combined 18 years of pharmaceutical experience with experiences GMP, development of small molecules, monoclonal antibodies, ADCs, vaccines, plasmid, combination products, and cell therapies. Looks like, David, you have done it all. And throughout his career, he has had direct experience with 25 products spanning the life cycle from discovery to post-commercial retirement. Dave has led roles from hands-on manufacturing process development, analytical development, qualification validation, project management, and quality system design. Prior to joining Kite, he worked at the MedImmune and Wyeth. So I would like to first congratulate Dave on yesterday's announcement of the approval of Tecartus. Congratulations. And Dave, can you share with us about a couple of one success stories and how that success becomes about?
Sure. Thanks for the congratulations. It's really great news to hear that. So as far as our success, I know it sounds cliche, but it's really, we focus on the patient. I know, we talk about cell and gene therapy and how it differentiates itself from the traditional biotech because once we get to large-scale manufacture, we're drawing a lot of people from that sector. And the ability to focus on the patient, because every single batch going out the door is for a patient, is really important. Where that kind of shows up is when we talk about time. So in traditional biopharmaceuticals, you'll make a batch and you'll get a deviation. You'll set that batch on the shelf and you'll spend a month to investigate and eventually go, okay, this is good or bad, and we'll disposition it, we'll send it out or reject it. That's not possible with the cell and gene therapy space. Once the patient is sat in the chair, and particularly in the autologous space where they're giving you their cells, you run off manufacture, you have very limited time to actually get that back to the patient. And that doesn't give us the opportunity in our quality systems to really kind of be set up the way you would in a traditional biotech sense. So we've got to adapt your quality systems to make sure that you're handling that time aspect, not just focused on the compliance, the quality, and taking as much time as you need to do so. With that, we also talk about kind of the material and how precious it is. So each batch that comes through our doors is an individual's material. So it's not a light decision if we say we're going to reject something and say we have to dump it and throw it out. It really is important. So again, kind of adapting your traditional CFR, ICH-based quality systems to include that medical end. You know, back in 90s, early 2000s, the small molecules, you'd never talk about having a doctor involved in a material review board to disposition a batch, but that's often the case with the cell and gene therapy space. And finally, again, focusing on that patient is differentiating this for your frontline workers. So it's really easy to get into a mindset when you're talking about traditional biotech of sending 100,000 vials out the door, they get put on a shelf, and you know, kind of they eventually get used, and people are excited about that because it makes a difference in someone's life. But really in the cell and gene therapy space, we're talking about literally it's in your hands, those are someone's cells. So having that impact is really important, and I think really helps people to focus on doing the right thing because they are focused on the patient.
Thank you so much, Dave. Really appreciate that insight and the patient-centered mentality. It's my honor to introduce the second panelist, Matthew Hobet, Dr. Matthew Hewitt. Currently, Matt serves as a Senior Director, Scientific Services, Cell and Gene Therapy at the Charles River Laboratories. Matt played a critical role in driving CGT strategic vision, as well as leading one of the Charles River CGT sites. Before joining the company, he was the head of R&D and clinical development for Lonza's personalized medicine business unit, where he led development of a cocoon manufacturing technology, a closed, automated, scalable cell therapy manufacturing solution. In addition, he developed and executed numerous collaborations across academia and industry that leverage the cocoon technology. Prior to Lonza, Matt led the tumor immunology and microenvironment program at the Bellicum Pharmaceuticals, which focused on improving cell therapy efficacy in solid tumors. He also led the immunology group at the UPenn gene therapy program, leading and contributing to numerous AAV gene therapy programs. It's a great pleasure to have you, Matt. Can you share with the audience one success story and how
Sure.
it become?
Yeah, so I think one kind of leads into the other. So certainly with my time at Lonza, we were focused on bringing the cocoon manufacturing technology to market and treating patients. Moving in the direction of closed, automated, and ultimately scalable manufacturing platforms is going to be one of the keys for cell therapy, specifically autologous or patient scale, to reach its full potential. In doing that and overseeing dosing of the first patient, first IND submission here in the US, began to uncover some other problems that exist in the field, and that's what I've taken with me a lot to Charles River. And I think what Charles River has assembled in an end-to-end solution for cell therapy and gene therapy development is quite compelling. And because now we have the capabilities to push the envelope a little bit on how we do things like manage critical raw materials, both for the supply chain as well as qualification, as well as, I think, nudge the regulators in the direction to speed up some of the ways that we do things like releasing various things, whether it's plasmid, vector, or cell therapies. Ultimately, you know, coming back to the original point, what this benefits is the patient. The faster we can move through development and into clinic, into more of a fast test, fast fail, hopefully fast success scenario, you know, the more we're going to benefit patients. And the failures are certainly as important as the successes, because we need to know what doesn't work as much as what does work. And to do that, we need a system which can keep up with us. And that's the thing that I'm seeing the most right now, is that We're not struggling, but I do think that there are some more efficiencies we can wring out of this system so that primarily the regulators can keep up with the innovators in the space.
Yeah, thank you. I cannot agree more. A end-to-end solution and a systematic approach to bring a quality and efficacious gene therapy to patients is of utmost importance. It's my pleasure to introduce our third panelist, Dr. John Rawley. John is the founder and the chief product officer at the Rooster Bio. John started Rooster Bio in 2013 as part of his personal quest of having the biggest impact possible on the commercial translation of technologies that incorporate living cells, including cellular therapies, engineered tissues, and tomorrow's medical devices. With a PhD from the University of Michigan in biomedical engineering, John has authored over 35 peer-reviewed manuscripts and 20 issued or pending patents related to biomaterial development, tissue engineering, and cellular therapy. He started his industry career at BD as a scientist and R&D manager in a cell and tissue technology group focusing on applying high-throughput screening technology to cell therapy media development and tissue engineering. John then contributed to the clinical development of Astron Biosciences tissue repair cell product, where he was senior manager of PD, responsible for manufacturing process improvement and cell delivery to the patient. John also spent 5 years as Director of Innovation and PD in Lonza Cell Therapy CMO business. Welcome, John. Can you share with us?
Yeah, I'd be happy to share with you one of the successes. And you know, my background is definitely in the process development side, and so you know, just like you know, everyone is talking about, it's all about, you know, getting these manufacturing processes developed and into the clinic as quickly as possible, right? So so that we can you know, try new, novel and innovative cell therapies in patients, right? And you know, back in you know, 2008 when I started at Lonza, which is a really great training ground, and you learn a lot. My job was to help you know, visualize and understand you know, the next generation MSC manufacturing processes. So the comments I'm making today are more on the allogeneic side as opposed to the, you know, personalized autologous type of products. And at the time, you know, Lonza was manufacturing for all of the major MSC products that were on the market. And as you want to get to that next scale, you know, the goal was to get the lot sizes from the you know, the low single-digit billions of cells per lot to eventually the tens of billions of cells per lot, and then the hundreds of billions of cells per lot. And when you actually go through the modeling and, you know, how many, you know, what is the dose size and how many patients are needing to be treated a year, you get to the math that a good go-to-market lot size is around 100 billion cells, about 10-fold greater than what the more very manual processes that were in 10-layer cell stacks or cell factories at the time were in. And then really a truly commercially relevant lot size was going to be in the hundreds of billions, if not a trillion cells per lot. And you need to get into 1,000, 2,000 liter bioreactors for that. Totally possible, and we started development of these bioreactor you know, manufacturing processes. And then as I was learning about the existing manufacturing processes that were going on in the suites, I got to gown up and go actually observe some of these processes. Actually realized that and the, you know, the manufacturing processes for MSCs at that time were, you know, between 40 and 60 cell stacks, these 10-layer vessels, right? And but that wasn't actually the issue. You know, the techs that were in the clean rooms could easily take down 80 to 100. However, where all the manufacturing bottlenecks at that time were in the post-harvest processing, right? And so you'd get 30 liters of cells and quenched trypsin, you know, out of these cell factories, and it would take hours to process them. And they were using, you know, some of the automated, you know, cord blood processing equipment at the time. And then even once they did have them processed and formulated in cryopreservation buffer, they were manually filled into cord blood bags, right? And so the manufacturing bottlenecks at that time weren't necessarily in the upstream. And so while we were focusing on bioreactors, if we did do a 10-fold increase, there's no way you're ever going to volume reduce and wash those cells. You're never going to be able to fill them into enough bags in the very short amount of time that you had. And so you have to take that entire manufacturing process into account. And then I was learning as much about the monoclonal antibody space as I could. I was from the cell therapy space, but Lonza had all these great sites, so I visited some of these other sites in Slough, UK, where they did all the great development, and learned about the automated and the scalable downstream processing technologies that we then tried to bring inside into Lonza and apply them to cell therapy manufacturing. Nothing was in blood bags; everything was in vials, right? So we championed and qualified technologies for switching from bags into pharmaceutical vials. They had to be special vials because you couldn't have glass vials for cells. They had to be plastic, and you had to fill them a certain way. But then once you made that transition to vials, you could fill thousands of vials an hour as you scaled up. And in the downstream processing, we developed TFF and eventually got to both a working TFF system and a continuous centrifuge, the KSEP technology that Sartorius eventually bought. And that was scalable to not just tens of liters, but hundreds of liters, and eventually thousands of liters. And so as the scaled upstream processing took place, you then had actually downstream processing at the same time. And I was telling this story about going to Bioprocess International for the first time, and there's a VP from like Biogen that was given a talk, and he said that 50% of his R&D budget was in downstream processing, as opposed to just the upstream. And so I modeled the R&D programs after that, and having those technologies done at the right time, because it's not just about scaling the upstream; it's also about scaling the downstream. It's just as important, and really balancing those things is really critical.
Excellent. Yeah, thank you, John. I think scaling up both upstream and downstream is the name of the game in the cell and gene therapy arena. So now we have a second round, which is to share from your personal experience, what is the lesson learned and what did you learn? Maybe we start from Dave?
Sure. So in terms of our successes, I talked about something very generic, focus on the patient. I'll get much more specific for our challenges, and it is selection of materials. So in the cell and gene therapy space, we're often taking something from an academic institution to GMP manufacture and trying to scale it. We see some type of clinical result, and very quickly it's make it commercial. That's very much a challenge because what happened was in the academic setting, someone took something off the shelf because they had it and they used it, and it was a research grade reagent, and that has now just stuck to the process. So it becomes very challenging to come up with consistent supply as you try to scale out. As you look for second source suppliers, this may have been a reagent that is only used very selectively, and there may not be second source suppliers. So it's very challenging to stick with the reagents that you have chosen, the materials, as you transition from the academic into the industrial setting. As part of that, you may also be using medical devices as part of your processing. John talked about using the cord blood apparatus, and you may use other things that are truly 510K registered devices. And you start to talk about to these people using them in your manufacturing process, and you want to start auditing them, you want to have agreements in place, quality agreements, all that kind of stuff, and they look at you like why do I care? You're not my client. My client is hospitals and those types of things. So it's a challenge as you start to put these manufacturing processes together with those materials. And what you end up doing then is taking all those materials and putting them into your regulatory filings. And you get questions back from the regulators, and you put more detail in your filing to answer those questions, and all of a sudden you have a commercial process that is very challenging to change any of those things, and you're stuck with a resource constraint in terms of being able to get enough of it in a consistent manner. So we have been very challenged with the materials we select, and I think the recommendation is as you very early on select what goes into your process, think about how that's going to scale. Don't hamstring yourself with picking something that is commercially available in a GMP manner, but definitely think about how am I going to get these out of my process or scale them out as we start to move through the clinical trials into a commercial setting.
Well said, Dave. I think that's quality design, not only from process point of view, but also from critical supply and supply chain risk management point of view. Yeah, thank you for that. Matt?
Yeah, I'd have to echo those concerns, obviously. I think, the other thing that we deal with, and the fact of the matter is right now, is that a lot of the innovations that are coming out of the academic space, so we are translating a lot of academic programs, sometimes that are preclinical, sometimes early phase, into something that's suitable for GMP and hopefully commercial. Certainly the reagents and security supply are a concern. I think one of the other things that we run into is moving away from the open manual processes, being one, into something that's a bit more automated, even semi-automated. The other being ensuring that the assays are in a form that they can be qualified and suitable for FDA. And I think this cuts across cell and AAV gene therapy as well. We've seen, I think, some pretty significant discussion on the AAV front about this in the last month or so, with the unfortunate deaths in the MTM1 program at Stellus, and formerly of Dentis, and then some of the other problems that we've had in some of the DMD studies. And there's some concerns around maximal dose as well as foreign protein that's being presented to patients. So, and on the cell therapy side, we've certainly had some issues in the TIL side of the equation with potency assays. So I do think that the lessons learned is going through your assay suite, especially if you're bringing in a program from an academic center, along with the reagents, fit for purpose equipment, to ensure that everything, can be actually made GMP and will stand up to the scrutiny of the regulators.
Yeah, so I have been serving on the CMC side to support over 20 R&D trials. I cannot agree more. So analytical testing, the qualification, validation, and the readiness for the agency is really critical, and cannot emphasize more. Thank you for that. John?
Yeah, the kind of the best practice and lessons learned, that I want to share today, from something that happened in the past that really set a program back is on the cell banking side. Again, this is specific for allogeneic cell therapies, right, where you create a master cell bank and a working cell bank, and these, developing, both the manufacturing processes and then building those cell banks can take quite a long time. They are big investments, and I was working on a project where we had developed a manufacturing process. We tech transfer that into GMP operations, and the project sponsor had decided to build their first cell bank based off a single donor because of mostly cost constraints. What happened was it takes time to tech transfer the manufacturing process, test it out. Actually, donor sourcing can take quite a long time, and then once you actually get that tissue and make your master cell bank, you've got about 6 to 9 months of safety testing that can cost well over $100,000 before that master cell bank is qualified for use for production. And everything was going fine, and again, took a few months to get the actually the tissue sourced and get that master cell bank made. And after kind of halfway through the testing, a positive result came back from one of the inapparent viruses that are this long list of viruses that are tested that the FDA requires in order to use. And there was a retest, and it actually came back negative, and so they thought it was a false positive. And after a couple months of just thinking about it, that cell bank was scrapped because that had a stain on it. Because if something did happen to the patient, you would always be wondering where it came from. And a good 9+ months were lost right? To save, probably, over $100,000 of sourcing that tissue and getting that master cell bank made. And then you had to go all the way back to the starting point and start back over. And really 9 months is an eternity in, for small companies that are trying to start up a clinical trial, right? And so really the best practice, from then on that I've always brought for me, and I always recommend to anybody, was, if you want to make one cell bank, you get at least 3 donor tissues. Even if you only want to test one at a time, it's everything. Every little piece takes time. But as much as you can parallel path, the amount of cash that you save is well by only doing a single one. Really isn't worth the time constraints that can put small companies in jeopardy that usually only raise money for 18 to 24 months. And so it really is always have that plan B, especially as it relates to your cell banks.
Yeah, so I really appreciate that candor, and I cannot imagine your blood pressure during those days. Yeah, so I also want to share from a gene therapy point of view, it's really critical to lay a similar solid foundation for cell therapy. Cell therapy side is good cell banking. On the gene therapy side, it's really critical to design a solid construct which is genetically stable and have a high productivity and also have a really good packaging efficiency. We have seen programs that the original gene of interest, the plasmid, is not stable or doesn't have a lot of a good feature, and so we have to rework the design. Then that put everything back. So that's a little bit sharing from my side. So we have 20 minutes left. As I said, we're going to make this panel, this world-class panel, available to the audience on site and the audience online for a free consultative session. And so I'll open up the floor. If you have questions on site, feel free to come up to the mic. If audience online, and we have our meeting organizer will read out your questions. So I'll now open up the floor. If you have questions, feel free to come up to the mic. Anything about CMC, cell and gene therapy.
Hi, good morning. My name is Anshika. I'm from the NIH, and I'm sorry if this is a naive question, but I have two-part question. One, like you mentioned that there could be batch-to-batch variation when you would do a small molecule manufacturing. Do we observe similar thing with the cell and gene therapy products? And secondly, I was wondering, how often do you see that the product or something that you bring from an academic lab doesn't pan out in an industrial side? Thank you.
Okay. So let me repeat the question for people who may not have heard. So the first one is regarding batch-to-batch testing variability and the manufacturing variability. The second question is regarding the academic assets that did not pan out commercially. Maybe we'll tackle the first one first. Who wants to go first?
I can start from allo, right? Which there should be a lot more. I was on an autologous company, and there was plenty of. Donor-to-donor variability is still by far the biggest manufacturing variability in allo specifically. Right? Once you move from one donor cell bank to another, there is tremendous variability in many of the quality parameters that we measure for mesenchymal stromal cells. And it's mostly in the potency area. But even once you have your cell bank and you have your working cell bank made and you launch a manufacturing process from one working cell bank to the next, there are tons of reagents that go into that process to expand those cells that can also lead to manufacturing variability. Now, if you got good control and good design around the media and the growth factors and that media can be made consistently with consistent levels of these growth factors, then you can minimize that variability. At the same time, the variability is also that you see is also dependent on the quality of the assays that you have, because there's just so much variability when it comes to live cells. And really the job of the manufacturing process developer, that's the job that I have, is to build a process that can accommodate as much variability as possible. But the most important thing is to understand what bad looks like, right? And when it doesn't work. And so that you can actually reject that lot or reject an entire donor before they go into that manufacturing process. You should not assume that once you have a manufacturing process design, that every donor is going to work for it, right? For MSCs, they're using such a wide variety of things. The best donor for cartilage regeneration might not be the best donor for graft versus host disease, right? Which is more of an immunomodulatory application. And so it is all about understanding what a good donor looks like, what a bad donor looks like, and screen those out before they ever enter manufacturing. So you definitely have to design a process to account for the manufacturing variability that you're going to see, and sometimes acceptable ranges for the quality attributes have to be big as you're optimizing that.
Yeah, maybe to add to that, I mean, I think what you're probably going to hear is that it's very modality dependent. So for MSCs, for instance, of course, potency related to secretome is always going to be a big question. And then, of course, you're going to have extracellular vesicles, and you're going to have questions about the origin of those versus are they from MSCs or are they from other feeder cells in the culture. I think in general, we break this into microbiological control and sterility, identity, and potency. And potency many times is really a surrogate of functionality, but potency is what the agency wants to hear. Because anything that we are testing in potency doesn't necessarily correlate directly to clinical efficacy. So you will see variability, but that's part of the process development, is to establish the relevant critical quality attributes of the product that you can meet consistently and reproducibly. And that's, again, from the agency has said, pick something, but pick something that is consistent and reproducible. It's not always going to be the same. Easy is always a relative term, but if we look at CAR-T, which is where the majority of the cells are at in the moment, MOA in that case is a bit easier to establish because you know what the cell should be expressing, and you should know what it should be killing based on that, in the case of oncology. But the identity is where this gets murky, is in some of the other modalities in oncology like TILs, where you have an unmodified TIL population that is multi-TCR, and you don't necessarily know what is actually doing what. And that's where you run into some problems around the classical, identity and potency assays, which you then have to create surrogates to. So we do see significant lot-to-lot variation. That's well known in allo and in autologous. Allo is potentially a little bit better, but I think, as was alluding to here, identifying the correct donors are key. We are seeing, in our cell supply business with HemaCare, we are seeing that those criteria for identifying donors is becoming much more complex. And that the reason why that's the case is because we're trying to, and working with the therapeutic developers, to limit some of the donor-to-donor variability we see in the allo products.
Thank you, Matt. We'll wrap up at the cell therapy side by Dave.
This one has come from I'm sorry, are we still?
Yeah, Dave has not provided.
So I appreciate John and Matt's contributions, because I think they took the really difficult question with how much variability is there, and the only answer I have is a lot. You talk about the variability in the assays, the variability in the reagents to get the assays, the variability in the manufacturing process. It's rampant, I guess. The second question may be about the success in terms of moving from academia to industry. I don't have the stats off the top of my head, but again, my answer would be a lot. I think there's a lot that kind of happens in a very small setting, and then when there's triggers to see actual work, it grows internally first before it actually moves to an industrial setting. So I don't have those stats off the top of my head. So I don't know if you guys.
I mean, certainly NIH has got some successes under their belt, right? I think academia in general does. They're becoming more savvy all the time. Many of the larger centers have their own GMP facilities, which they run early phase clinical trials through. Always the key there is that typically the academic centers don't want to be the IND holders for any pivotal studies. So then they have to move out into, another entity, whether it be somebody like Gilead or otherwise. Typically, we deal in CDMO world with a lot of the academic programs coming in, and I would say I haven't seen too many that quote unquote failed to the point where we couldn't get it to work. But I mean, I think that's a process question versus a clinical question.
Yeah, I want to address the lot-to-lot variability for viral vectors. We typically approach this this manufacturing control from 5M, so manpower, method, measurement, machinery, and material. So every single M can contribute to the variability of the manufacturing. Especially for phase 1, preclinical phase 1-2, because those processes are yet to be validated. So it's typical to see variability lot to lot for manufacturing, from manufacturing point of view. And your second question for the because on the Vigene Biosciences, we have actually research productions. We have seen a lot of academic assets move into clinical trials and pick it up by commercial companies. So we have seen a lot of success. Yeah. Okay.
This question comes from the online audience. At your companies, are you doing in-house R&D or reaching out to CROs? And what about manufacturing and cGMP operations?
So this question is, so our panel, I think this question probably primarily for Dave, because the others are CRO and the CMOs. Dave?
Yes. So, I mean, we are doing our own R&D in-house. We have our own GMP manufacturing facilities to do both clinical and commercial. We are partnered with academic institutions and other companies. All of that is, publicly researchable. But, one of Kite's superpowers, I guess we claim, is the manufacturability. So that's where we're looking to be some of that, not CDMO, but to work with some of the companies that have the technology out there and leverage our ability to manufacture.
Thank you, Dave.
And I mean, I would, as a small reagent supplier, right? So we are a GMP reagent supplier, but we're actually too small to have our own GMP, manufacturing at the time, or GMP analytical assays. And so while we develop all of our manufacturing processes, we have internal R&D. We are dependent on GMP manufacturers and GMP testing houses to make and release our products. At some point in our business model, we will bring all of that in-house, but it takes time and growth, right? So but at our stage, and we're about a 45-person company right now, we couldn't keep a really expensive GMP operation, functional year-round. So.
Right, and maybe I'll add, Charles River is a little bit bigger, obviously, but a little bit, yeah. I mean, we're classically known for our animal models as well as safety testing. In the last 2 or so years, we've acquired a number of companies in the CGT space where now we can really take somebody from concept to commercial, including antibody discovery within CGT. So we do have significant capability, certainly in R&D for clients. We have certainly an arm internally where we do R&D to increase the stable capabilities of Charles River. And then as part of, Vigene, Cognate, Cobra, Hemocare, we certainly have the ability to do GMP tissue acquisition, from donors, plasmid, as well as vector manufacturing and ultimately cell therapy manufacturing. And at Cognate at the moment, we have clients that span the realm of divides here from early phase 1 all the way through commercial readiness preparation. So PPQP, PLI, PAI. So we have various capabilities at our disposal.
Thanks, Matt. Thanks, John. All right, we have 7 minutes left. Any other questions? Yeah, come on over.
Hi, I'm Tom Genova. I'm from IDT Biologica. I wanted to understand, with COVID-19, there's obviously been some CMC hurdles that you guys probably had to jump through. I wanted to see how you're addressing some of the supply chain issues related to raw materials to be able to keep projects from going off the tracks.
Thanks, Tom, for that question. So supply chain is definitely seriously disrupted and strained by the COVID. So I will first let our panelists address, and I will chime in.
So yeah, Kite, being a continuous manufacturer, has a certain amount of safety stock of its raw materials, and at the very beginning kind of forecasted and looked, what's going to be problematic, and looked at what are the longer lead items, and do we want an additional safety stock of those, and just kind of, again, in that continuous risk management mindset, look at what did we need more of to keep in-house, and what is evolving. So what are we hearing from our suppliers in terms of how is their lead time changing, and how do we adapt to that? So kind of that continuous mindset of just, as we get feedback from our suppliers, changing on a individual supplier basis.
Yeah, I've dealt with this in a couple different areas. One, certainly being reagents. I think in general, obviously shutting the entire supply chain down had a major shock on the system, and then trying to open it back up and everybody kind of scrambling to then place a bunch more orders for safety stock has compounded some of the issues, as well as some of the NDA effects of sourcing stuff for COVID-19 vaccines. Because a lot of the materials we use in manufacturing are many of the materials that are used in mRNA and vaccine manufacturing. But in general, we've identified secondary suppliers, of course, placed orders. We're in continuous contact with our suppliers to get partial release of an order to us so that we can continue operations. And much like everybody else, just in this field, I think just because COVID happened doesn't mean that cancer stopped or any other ailments that we work in CGT. So, our folks have been working throughout the pandemic, and, we've been very appreciative of that fact.
Thanks. Ditto. Yeah, it's all about strategic supply chain, having someone think about it all the time.
Yeah, I would like to add just to partner with the vendors and the suppliers on the both high level and on the also operational level, to let them see your need and have advanced supply and even some blanket PO. Just let them see your need. I think that will go a long way. Okay.
I think this is a follow-up to the... Oh, I'm sorry, you go ahead.
Hi, Randy Rovato, Sci PhD. Kind of related to supply chain. I'm actually interested in the workforce supply chain. So in CMC and in operations, that kind of thing. What's the relative role that you see for the PhD level talent versus the, not lower, that's not the right way to say it, but where is the role and the need and the ability to retain the PhD level talent in the CMC and the operations type versus the more traditional research side?
Great question. So I will first open up to our panelists.
Yeah, I think this is something we're probably all struggling with on some level. The field has grown much faster than the talent pool that's available, so we are all the time constantly reevaluating the way that we're going out and hiring, as well as the qualifications we're looking for. Especially in the I hate to say it, lower roles, but in some of the traditional manufacturing associate roles, we're looking at whether or not we actually need certain levels of degrees, as well as doing more personality fit, because a lot of this work can be done with we can train people up. So that's what we've been looking as more long-term development of folks, as well as partnering with academic institutions to create funnels into our sites.
Yeah, I usually don't see PhDs going into manufacturing, like right out of school, they wouldn't take an entry-level manufacturing job. I have seen lots of PhDs successfully come into R&D, get into process development, really learn how to develop manufacturing processes, and then move into manufacturing in a more or quality in a more management role in manufacturing, or in a strategic CMC role on the quality side. So, but yeah, I typically don't see them in fresh out of school just going and working in a clean room. That routine work is usually not of interest to the PhDs. So.
Right.
Yeah, I guess I'd say specifically PhD level, it's an issue resolution. So, go back 20, 30 years into small molecule manufacturing, not a lot of issue resolution, not necessarily needing a ton of expertise, but move forward to the amount of variability we talked about earlier in terms of every single batch, and there's lots of issues. So in a tech services function where they're helping to troubleshoot something went wrong, and back to material being precious and trying to get it to a patient no matter what, we can see some of that more higher level thinking rather than the frontline manufacturing that John's talking about.
Yeah, I'd like to chime in with my thought, because I'm PhD trained, but very little did I learn from my graduate school days that I can use in my current role. I think PhD level scientists definitely play an important role in CMC manufacturing. We have a couple of areas can definitely utilize PhD trained scientists: process development, assay development, project management, and some of the business development, because also can use PhD trained scientists, because their counterparts on the client side are all PhD trained. The knowledge itself actually is less important than the system and the critical thinking that we developed during the graduate school days. So that would say that's most important. Okay, I don't know if our organizer, okay, I'm giving the sign that we need to wrap up. Yeah, I would like to, first of all, thank you for your attention, and the people online, thank you for listening in, and also now join me to thank our panelists for their great expertise.